Hi guys, today we are going to see about production of food yeast and fooder yeast, a comprehensive guide to industrial single cell proteins (SCP) productions. What is yeast biomass? Food yeast produced specifically for human consumptions. It is inactive, safe, and rich in proteins and vitamins. Typically grown on molasses or defined medium to ensure high purity and quality. Another one is fooder yeast. Yeast biomass used as a protein supplements in animal feeds. It often utilizes cheaper substrates like sulfate, waste liquors, or wood hydrolyzate, turning industrial waste into valuable proteins. Key microorganisms Saccharomyces cerevisiae, primary species for food yeast, bakery, nutritionals, high quality of flamers for food industries. Candida utilities common for fooder yeast, can utilize pentose sugars found in wood waste and rubber growth. Raw material for molasses can and beet molasses, the most common substrate for food yeast productions, rich in source, glucose, and fructose. It requires clarification to remove calcium sludge and colloidal matter before use. Provide biotins, vitamin B7, which is essential for yeast growth. Raw material for sulfur liquors, paper industries byproduct. Waste effluent from the sulfate pulping process of oats contain fermentation sugars, exose and pentose. Candida is performed here as it can metabolize pentose sugars that Saccharomyces cerevisiae cannot. Must be stripped of sulfur dioxide SO2 before fermentation as it is toxic to yeast. Raw material cheese whey, dairy waste utilization, liquid byproducts of cheese manufacturing, rich in lactose, which constitute a pollution hazard if dumped, requires specific strains like Clebsam mexicans, family key processes which produce lactase enzymes, produce a high quality food grade yeast biomass. Production overview. General step up. Upstream strain preservation and media preparation, fermentation, aerobic growth in bioreactor, downstream process, separation, washing and drying. The process transforms low value carbohydrate into high value protein via biological conversion. This is the overall picture to explain the upstream of fermentation and downstream fermentation. Upstream strain selections. Criteria for selection. Strains are selected based on genetical stability, growth rates, generation times, and the ability to utilize a specific substrate. Restraint to contamination and high temperature tolerance are also favorables. Inoculum preparations. Start in a test tubes, moving to flask, shaking flask, and then seed fermenter. This scale-up process ensures main bioreactor receive a healthy, active, and sufficient large population of yeast cell. Media preparation. Clarification. Raw materials like molasses are diluted and heat treated to precipitate impurities which are then removed by centrifugations. Supplementation. Nitrogen source, ammonia and urea and phosphoric acid are added to balance C into 7:P ratio. Sterilization. The media is sterilized using HTST, high temperature short times, continuous sterilization to prevent contamination. Laboratory scale up. For lab to plant, quality control begins here. The purity of this culture is checked microscopically at every stage. A small contamination in the lab's flask can result in a massive failure in the 1 lakh liter fermentation tanks. The bioreactor. Heat of productions. Industrial fermentation takes place in large stainless steel tank, equipment with cooling jacket and aeration systems. The process is strictly aerobic. Unlike alcoholic productions, we went to maximum biomass, so high oxygen levels are maintained to support alcoholic forming pasteurized effect. Critical parameters. Temperature. Optimal 30 degrees Celsius cooling coils are essential as yeast growth and generate significant metabolic heat. pH level maintained at 4.5 to 5.0. This slightly acidic environment favors yeast growth while incubating many bacteria. Aeration. Vigorous aeration 1 ppm is critical. Oxygen transfer rate is often the limited factors in productions. Food yeast production specifics. Feed method. Fed batch culture is used to keep sugar concentration low 0.1% to prevent the carbonate effect. Alcoholic production. Substrate quality. High grade molasses or glucose syrups is used to avoid heavy metals or toxins. Washing. Extensive washing is required to remove molasses color and taste. Goals: high protein content, pleasant flavors, and vitamin B complex enrichment. Fooder is production specifics. Continuous culture, often produced in continuous systems to maximize throughput and efficiency. Substrate flexibility can tolerate deteriorated substrate like sulfuric liquids or agricultural hydrosolids. Yield focus. The primary goal is maximum biomass yield per unit of substrate. Harness Candida species used are generally more resistant to inhibit the found in waste substrate. Food, food yeast versus fooder yeast. Features: food yeast and fooder yeast. Primary organism Saccharomyces cerevisiae, Candida hydrolysis. Substrate for food yeast molasses and glucose. Substrate for fooder yeast is sulfuric and liquid wood hydrosolate. Protein content for food yeast 4.5 to 5.55 percentage. Protein content for fooder yeast 40 to 50 percentage. Processing: fooder yeast extensive washing and mild drying. Processing: fooder yeast standardized washing, drum spray drying. Uses: food yeast used human consumption, fooder yeast animal feeds. Harvesting: centrifugations. At the end of the fermentation, the yeast concentration is absorbed 4 to 5% solid. This start centrifuge are used to separate the yeast cells from the spent mediums. This concentration the yeast creams is to about 18 to 20% solid. Drying methods: spray drying, most common for fooder yeast. Atomize yeast cream into hot air, produce the fine light powders. Preserve yeast sensitive vitamins. Drum drying, often used for fooder yeast. Yeast cream is dried on hot rotating drums, producing flax or sheeter, which are then grounded. More energy sufficient but higher heat exposure. Nutritional composition: dry weights. Proteins 50%, carbohydrate 35%, mineral 8%, lipid 5%, liquid acid. This overall comprehensive protein size higher than the other component. Yeast is an extensive rich source in complex protein and B complex vitamins. Application: food industries. Nutritional yeast. Solid flask of powder. It is a staple in vegan diets for its cheeses, nutty flavors, and B12 fermentation. Flocculation. Flavor enhancer. Yeast extract are used in soup, sauce, and snacks foods to provide umami flavor. Slower taste replace MSG. Application: animal feeds. Poultry improve growth rates and feed efficiency. Serve as a probiotic to improve gut health. Aquaculture replace expensive fish meals in diet for shipping and salmon. Cattle and pigs provide essential amino acids like lysines that are often limiting in grain-based food feeds. Economic importance: market growth. The global yeast market is projected to reach over $8 billion by from 2026. The demand for alternative proteins and clear label ingredients is driving this expensive. Cost effectiveness: fooder yeast convert low value industry waste, sulfur and liquid whey into high value protein, considering creating a critical circular economic. Sustainable waste to waste wealth. Economic impact: yeast production reduce environmental pollution by consuming high BOD biological oxygen demand waste from paper and dairy industries. It requires significantly less land and water than traditional animal farming to produce the same amount of protein. Challenge in productions: high nucleic acid content. Yeast contain high RNA 6 to 10%. High consumption can lead to uric acid buildup in humans. Processing it needs to reduce this for food grades. Contamination: maintaining a sterile environment in massive bioreactors is technically demanding and energy intensive. Heat generation: massive cooling costs are increasing to maintain the 30 degrees Celsius optimum temperature. Thank you.